So our project was focusing on the microbiology, and here we asked the question if biodiversity can act as an ecological barrier to the spread of clinically relevant antibiotic resistance in the environment. So as you saw, we were quite a large consortium, and I just briefly want to introduce why AMR is important. There was an article which I think described it quite well because it called it the silent pandemic. Silent pandemic because you can be colonized by an antibiotic-resistant bacteria and you will still be healthy and you will not notice it. But you will run into problems as soon as you get ill and the doctor tries to treat you with antibiotics, and then you are colonized by the resistant bacteria, which may transfer their resistance to pathogens in your gut, for example. And as you see here on the left-hand side, this is expected to increase dramatically. Now, there's a strong environmental aspect of AMR, which is on the right-hand side illustrated. It was a large consortium project where we also were participating, where you see that we more or less in our sewage have everywhere releasing AMR. Bigger problems we have in South America and Africa, but also in the countries which have a higher BIP, you see that we do release quite a lot of different combinations of AMR. Now this is empty on my scale. Okay. Our hypothesis, therefore, was if diversity can serve as a barrier, microbial diversity in this case, that is, can serve as a barrier to the invasion of antibiotic-resistant bacteria and their transferable antibiotic resistance genes. And therefore, we analyzed if diverse environmental communities contain less resistance genes than less diverse communities. For that, we analyzed pristine soil and river environmental samples across Europe. Pristine means less impacted or less disturbed. So pristine is a bit of an exaggeration. And as I said, we analyzed aquatic ecosystems and soil ecosystems and analyzed the 16S DNA to get an indication of the microbial diversity, and then used, with the help of resistomap, an analysis of a large amount of resistance genes. Overall, we had 200 samples. We did this in a replicated manner. So we, in each of the participating countries, we analyzed forests and aquatic, or soil and aquatic systems. And this is the main result, which we have also already published. And here you see two graphs. On the top is the river, and on the bottom is the soil. And one surprise for us was that we didn't see an effect in the river, but we saw a clear effect in soil. So if you see with a look at the lower panels, with an increasing diversity, we have a decrease of number of resistance genes. So there, this hypothesis could be supported for soil. If we have a higher diversity, we see a lower number of resistant genes present. I know. And this has, of course, already implications, for example, in the context of water reuse, which is currently discussed a lot. And there we try to apply these results already in terms of having a more healthy soil, and if that can be better used in the context of water reuse when you use treated wastewater for irrigation. And with that, I should be in time, and really want to say a big thank you to our ANTIVERSA team. It was a great pleasure to work in this team, and I hope we can continue this in some other manner. I'm sure that's true for many other projects as well. Thank you very much for listening.